Because I said what was on my mind. I made a lot of enemies. I made a lot
Welcome, everyone, to Science from the Fringe. My name is Bryce Nickels. I'm a professor of genetics at Rutgers University, and today I'm joined by my colleague and longtime collaborator, Richard Ebright. Richard is a professor of chemistry and chemical biology at Rutgers, and like me, runs an NIH-funded research lab at the Waksman Institute of Microbiology. He's internationally recognized as a leading voice for effective oversight of risky pathogen research, so-called gain-of-function work, and for his advocacy of transparency and accountability surrounding the likely lab origin of COVID-19 and the efforts to conceal that truth from the public. These are issues that I, of course, got heavily involved in myself over the past 3 years because of Richard. But today's discussion is going to be something much different than I think most people are used to Richard talking about. And that is, we're going to talk about Richard's own research and discuss how that ties into Richard's strong opinions in another area of science policy, one that I think is a little less polarizing than discussions around gain of function, despite it being listed as one of the top 10 public health threats facing humanity, at least according to the WHO, and that is the growing threat of antibiotic-resistant bacteria. So, Richard, thanks for joining me today. How are you doing on this rainy Sunday afternoon in Central Jersey?
Fine.
That's it? That's all I get? I mean,
Weather question?
weather question, this is how you run podcasts, Richard. Maybe you are not familiar with the way that professional podcasters run the show, but this is the way we do it. Okay, so are you actually excited to be talking about something other than COVID and gain of function?
Of course.
Okay, so can you tell me about your research program? Like, just give, in general terms, what is your research focused on?
We perform research in two areas. About half of my laboratory's research focuses on defining the three-dimensional structures, the atomic structures, and the mechanisms of action of the molecular machines that bacteria use to synthesize RNA. For that work, we use tools of structural biology and structural biophysics, tools that enable us to define the full atomic structures of the large molecular complexes, the large molecular machines responsible for RNA synthesis, and that enable us to see the changes in those molecular machines that occur reaction by reaction, step by step, as they carry out RNA synthesis. So that's about half of our effort. The other half of our effort focuses on antibacterial drug development. In that part of our work, we seek to use information that we and our colleagues have developed from structural and mechanistic studies of the molecular machine that synthesizes RNA in bacteria to identify new compounds that interfere with the action of that machine, and by doing so, kill bacteria. These are compounds that can be developed into new antibacterial drugs, new antibiotics.
How long have you been working on antibiotic discovery?
We've been working on studies of RNA synthesis in bacteria in my laboratory at Rutgers since the laboratory opened in the mid-1980s. And we've been working on translating our findings into new drug development, new antibacterial drugs, since about 2000.
I do recall that you got into antimicrobial development right around the same time you started getting involved in the gain of function debate. And
That's correct.
in fact, we'll come back to that later because I think it relates to the amount of funding that's in, research in this area versus research in virology. So, how many compounds has your lab developed?
It's probably better to start with how many targets we've worked on. So our fundamental target, molecular target for drugs, antibacterial drugs affecting RNA synthesis, is RNA polymerase, the molecular machine that bacteria use to synthesize RNA. When we seek new compounds that inhibit RNA polymerase. We're seeking compounds that will target parts of the RNA polymerase that are conserved over multiple different species of bacteria, because we're interested in having compounds that will be able to kill multiple different species of bacteria. And we're seeking targets within RNA polymerase that are not conserved, not similar in the RNA polymerases in mammals, particularly in humans, because we're seeking compounds that will kill bacteria but will not have effects on human RNA polymerases. And most importantly, we're seeking targets within RNA polymerase that are different from the targets of currently known antibacterial drugs that function by inhibiting RNA polymerase. And there are two classes of currently used antibacterial drugs, one called the rifamycin class, one called the lipomycin class, or fidaxomicin. Both of those classes kill bacteria by inhibiting the molecular machine that synthesizes RNA. But for both of those classes of antibacterial drugs, there are strains of pathogenic bacteria. They're bacterial pathogens that have become resistant to the drugs. And resistance arises almost always from mutations that alter the binding site on RNA polymerase for the drug. So when we look for useful targets within RNA polymerase, we're looking for targets that are different from those that are the targets of the currently used drugs, where there already are mutations within the molecular machine RNA polymerase that confer resistance. So over the course of our work in the last 2.5 decades, we've identified 7 different potentially useful targets within RNA polymerase, 7 parts of RNA polymerase that perform key steps during the process of RNA synthesis that are conserved among many different species of bacteria, that are not conserved in human RNA polymerases, and that are different from the targets of currently used drugs that function through RNA polymerase as a target. For each of those 7 targets, we've carried out systematic search. We've carried out systematic searches, screens, high-throughput screens of microbial culture libraries and synthetic compound libraries, seeking new compounds that inhibit RNA polymerase through the new target. And for each of the 7 targets, we've identified at least one hit, at least one compound class that inhibits the enzyme through the target. For each one of those new compound classes identified, we've defined the details, the structural details of how the compound interacts with RNA polymerase by determining atomic structures. By determining atomic structures of the compound bound to RNA polymerase, we've characterized the biochemistry of inhibition, the mechanism of inhibition. We've characterized the profile of which bacterial species are inhibited by the compound. And in favorable cases, we've demonstrated antibacterial activity not only in culture, but also in animal models of infection. Out of that work, we've identified 4 sets of compounds that we currently focus on. 3 of these sets of compounds are compounds that target mycobacteria, the group of bacteria that cause tuberculosis, as well as important additional infections called non-tuberculous mycobacterial infections. For one class, we've identified compounds that target a very broad range of bacterial species, and we focus on those compounds, seeking to develop those as treatments for lower respiratory tract infections and skin and soft tissue infections.
So it sounds like you actually run what could be described as a mini pharmaceutical company in your lab.
The part of the laboratory that does translational research is doing the kind of research that traditionally has been performed in big pharma by large teams in pharmaceutical companies with large teams and large budgets. But as we'll discuss over the course of this podcast, large pharma, big pharma, has left this discovery space, no longer is operating in this discovery space. And so any effort to develop new antibacterial drugs today is being done by small companies and academic labs.
So, and how many patents have you generated out of your work?
I think we have 50 issued patents, or approximately 50 issued patents. those cover different areas of research, but the majority of these are in antibacterial drug development. So probably 35 or 40 issued patents, as well as additional patent filings.
How much money have you gained personally through this research? Have you generated any revenue yourself?
In terms of personal income, almost none. And again, as we'll discuss further in this podcast, when Big Pharma left this space and when investment funds left this space, and when a market failure occurred that has prevented investment funds from re-entering this space, this becomes an area of research that one does because it's important and because it's beneficial. This is not an area of research that one does in order to boost one's income. It simply doesn't happen.
And that's a point I'd like you to address, actually, because I do find that it is, at least personally, difficult to explain to people how to distinguish scientists that have vested interest and the financial vested interest in the research they're doing, that's driving some of their positions, and ones that are, communicating the actual urgency in a manner that is real. And this gets to going back to, pandemic preparedness and how we saw COVID emerge. Could you maybe make the case that what is the threat of antibiotic resistance, or what's the best term? Antimicrobial resistance or antibiotic resistant bacteria? What do you want to call the threat?
Policy specialists generally refer to antimicrobial resistance, or AMR. But to a very good first approximation, antimicrobial resistance, AMR, means antibiotic resistance.
So we saw the virology community, actually we see it to this day, constantly talking about the threats of pandemics and how millions of people will die if we don't urgently do stuff like give them more money. Why? What's the case made for combating antibiotic resistance? Why should more money be invested in that? And is the threat of antibiotic resistance, I guess the question here is, how can the members of the public tell that when you say, all right, millions of people are going to die of antibiotic resistance, how does that make you different than Peter Daszak, who stands up there and says, there's these imminent threats and that sells, we can get lots of grant money?
Is that this is an issue of equity, and whatever we try and do to think this through, there's a huge equity problem. Now, as we go to politicians to talk about our agenda, I think I've got a much easier sell to a funder or to a politician on, we're going to stop the next pandemic if you give us, a couple of $1 billion, we'll fix it. But the hype and concern is real enough to drive huge amounts of money for that issue. And, and speaking more dramatic languages about threats. We talk about global health security. I mean, that's a great sell. So I think there's an equity issue that can be used to sell this agenda in the richer countries and use that funding to really deal with these issues.
Make the scientific case for that, but then also, is there a way for people in the public that are sort of looking at people that are communicating different threats? Can they distinguish who's telling the truth, so to speak, and who's just a bunch of hype and bluster?
One needs to look at statistics, actual facts, namely the burden of disease now, the predicted burden of disease in the future. And one needs to look at that at the level of the globe, and one needs to look at that at the level of the United States. So let me summarize the numbers for antimicrobial resistance, antibiotic resistance. Currently, each year, more than 1 million deaths occur worldwide that are directly attributable to antimicrobial resistance. That is to say, that are caused by antimicrobial resistance. And there are an additional 4 to 5 million deaths that are related to antimicrobial resistance, where that is a contributing factor, but not the cause per se. This is a large death burden, but it is an increasing death burden. The numbers have been increasing, and the World Health Organization estimates that by 2050, the numbers will be double the numbers now. That is to say, 2 million deaths annually directly attributable to antimicrobial resistance, and up to 10 million deaths that are related to or contributed to from antimicrobial resistance. That's at the global level. There also is an enormous economic burden associated with those numbers. The World Bank estimates that if current trends continue, bacterial drug resistance will increase global healthcare costs by up to $1 trillion per year in 2050 and will decrease the global gross domestic product by up to 3.8% per year by 2050. Those are global numbers. The numbers in the United States are also significant. Currently, each year, the CDC estimates that 36,000 persons in the United States die from drug-resistant bacterial infections. And the estimate is that those numbers will scale considerably over the next 3 decades.
Do we trust those numbers, though? Here's the problem. Do we trust the numbers from the CDC? Do we trust the numbers from the WHO? Can you convince people that those numbers are not inflated? I do think there is a problem now where when you have real disease threats that the public doesn't trust the numbers that are used to explain that. So how can, I mean, is there additional evidence that those numbers are correct, or?
The numbers don't come only from those agencies, the WHO, the World Bank, the CDC. Those agencies are distilling reports, statistical reports that come from the United States government, that come from state governments on reportable diseases, and come from academic laboratories of epidemiologists surveying the statistics. And there is complete accord across the board at the level of national statistics, state statistics, academic reports, and global and national organizations like WHO, World Bank, and CDC. There is no basis to doubt these numbers. There's no controversy in these numbers.
So maybe let's back up for a second. Can you explain how antimicrobial resistance develops and what are the current drivers of antimicrobial resistance or antibiotic resistance?
Okay, so first we start with what is antimicrobial resistance or what is antibiotic resistance? Microorganisms that are treated with drugs, like bacteria treated with antibiotics, are able to acquire resistance. They evolve resistance as a result of direct selection of resistance. And the important thing to know in understanding why this occurs and why this occurs quickly and why this occurs inevitably for any compound is that in a microbial infection, particularly in a bacterial infection, the number of infecting organisms in a single patient is enormous. So in a patient with an infection, it is not unusual for that patient to have 10 to the 8th, that's 100 million infecting pathogenic bacteria, up to 10 to the 9th, that's 1 billion infecting bacteria. It is also not unusual for those bacteria to have life cycles on the time scale of tens of minutes, for example, 20 minutes per generation or 30 minutes per generation. When you have hundreds of millions of organisms replicating, reproducing on the time scale of tens of minutes, and then you treat them with a compound that kills those compounds, that provides an enormous selection over enormous populations with short generation times to evolve a resistance. Every antimicrobial compound that has been discovered to date after use in the clinic in humans has given rise to resistance. The microbes have developed mutations that allow them to overcome the drug. This can happen in a number of ways. One way that it can happen, the simplest way that it can happen, is simply for a mutation that changes the target molecule inside the bacterial cell so that it no longer binds the antimicrobial drug. It can also happen in more complex ways where the microbe actually acquires the ability to degrade the drug or to export the drug from its cell or to prevent the drug from getting into the cell. And it always happens. Every compound that has been discovered when applied in the clinic on the time scale of years to decades has resulted in antimicrobial resistance such that the compound no longer is effective against the bacteria that are present in the population, in the community, globally, and available to infect. So that's antimicrobial resistance. And the key things to keep in mind are that it occurs relatively quickly on the time scale of years to decades after a compound is introduced into the clinic, and it occurs inevitably. It will occur with every new antibacterial compound. That means that the only way humans can continue to be in an antibiotic era, an era where we have those tools, those drugs that are absolutely essential for modern medicine, absolutely essential for transplant medicine, absolutely essential for treatment of cancer and cancer patients, absolutely essential across the board. The only way we can keep them is to keep developing new ones, because each one that's discovered and developed and introduced into the clinic within 5, 10, 15, or 20 years will need to be replaced by a new compound where the bacteria have not seen it before and are not resistant to it, but now will begin the same cycle, necessitating replacement of that second compound in 5 to 10 to 15 to 20 years.
Is there a way that you could explain how antibiotic resistance, the mechanism by which that occurs, how does that relate to how vaccines can lose their effectiveness over time?
We all have seen during the last 5 years that vaccines against COVID that were introduced at an early stage drove resistance to the vaccines in an analogous, not identical, but in an analogous fashion, such that rare viruses in the population that had mutational alterations that moved them away from the viral forms that were targeted by the initial vaccines acquire a growth advantage and come to dominate the population of viruses in the community, the global population of viruses, causing the vaccines to lose effectiveness with time. So there's a similarity, but it's a more direct cause-effect relationship when you have a bacteriostatic or bactericidal compound. That's a compound that causes bacteria to stop growing or to actually be killed. There's a direct selective pressure then for the bacteria to acquire resistance.
Can you go over the bacteria that present the most, the largest threat to the public?
The 8 sets of bacteria that cause the largest burden of disease, a burden of disease that represents 11% of global total disease from all causes. And that set of 8 bacteria, those eight sets of bacteria include the mycobacteria. That's the group of bacteria that cause tuberculosis as well as non-tuberculous mycobacterial infections, the streptococci, the staphylococci, genus Escherichia, genus Klebsiella, genus Pseudomonas, genus Enterococcus, and genus Acinetobacter. Those 8 collectively destroy almost 300 million DALYS, again, making them the cause of about 11% of total global disease.
What are some of the societal practices that contribute to antimicrobial resistance?
So from what I've told you already, I've told you that when new antibacterial drugs are introduced, that begins the process of driving a process of resistance. That resistance can occur in humans when the drugs are administered to humans. That can also occur in livestock when the drugs are administered to livestock, and that resistance can occur in crops when the drugs are used in treatment of bacterial infections in crops. The largest use by ton is in livestock. Livestock tend to be large-bodied animals. They take large doses, and antibiotics are used routinely in much of the world, especially in the United States, as growth promoters, as routine feed additives to promote growth of livestock.
Why do they promote the growth?
It's not entirely clear, but by eliminating bacterial infection, that reduces stresses on the animals. That's part of the explanation, but probably not the entirety of the explanation. But so use in the clinic, in human medicine, use in agriculture, both in livestock and in crops, those provide drivers. Those are drivers of resistance. And then when we consider use in the clinic for infections that require the antibiotic represent good uses, but there is considerable overprescribing of antibiotics, and each use of antibiotics when there is not a necessity is driving resistance without a good reason.
When you talked about first getting into antibiotics back in 2000, that was right before there was the Biodefense, the large influx of money into biodefense that happened in the wake of 9/11. Did that influx of money into biodefense result in a reduction in money going to develop new antibiotics?
Initially, it did. So the immediate response to the 9/11 terror attacks in 2001 and the anthrax mailings in 2001 was a very large-scale prioritization at NIH, in the National Institute for Allergy and Infectious Diseases, NIAID, a very large prioritization of research funding for biological weapons agents. And the funding initially was tied to a list of NIAID category A, B, and C prioritized pathogens. So there were specific lists of the bioweapons agents of highest concern, of second highest concern, and of third highest concern as categories A, B, and C. And eligibility for prioritized funding was linked directly to research on those agents. That began in 2003, reached its high point perhaps by the end of the Bush administration, 2007, 2008. After that time, the prioritization of category A, B, C agents continued, but now also funding was provided for antimicrobial resistance that was not being funded at a high level initially.
So I guess that leads into the policy part of this. Is your contention that there's enough money that has been invested in the development of new antibiotics, but it's bringing those to market that is the current problem?
The real bottleneck currently is a market failure that occurs after the science and separate from the science. But on the question of research funding for antimicrobial resistance and new antimicrobial drug development has not kept pace with the scale of the disease burden, the medical impact of antimicrobial resistance. Some simple numbers just explain that. Right now, 30% of all infectious diseases death in the U.S. is due to antimicrobial resistance. It's from the subset of microbial infections, of infectious diseases, of bacterial infectious diseases, and fungal infectious diseases that are resistant to the current drugs. So 30%. The institute at the National Institutes of Health that's responsible for infectious diseases, NIAID, budgets 8.6% of its budget for antimicrobial resistance. And so 30% of the death impact in the U.S., but 8.6% of the funding. So it is, relatively speaking, underfunded. It's particularly underfunded relative to certain other areas. HIV/AIDS is responsible for just 3.8%, so less than 4% of U.S. infectious diseases deaths, but it receives 58% of the NIAID budget. And so if we simply compare NIAID research dollars to U.S. deaths, HIV/AIDS receives 50 times as much money in terms of dollars per deaths as antimicrobial resistance. And that represents a political decision, entirely a political decision. Activists for HIV/AIDS patients, persons with HIV/AIDS, have driven major increases in spending in that area. There hasn't been a corresponding lobbying effort for antimicrobial resistance.
It might be useful to go back a little, well, not to go back necessarily, but talk about how new antibiotics are discovered. And of course, if you want, we can talk about the role that Rutgers University played in the history of antibiotics. Maybe we should do that right here. Do you want to talk a little bit about what unique place in the history of antibiotics Rutgers University plays?
The overwhelming majority, almost all of the antibacterial drugs, the antibiotics that are in current use in the clinic today, were discovered in what's referred to as the golden age of antibiotic discovery, which began in the 1940s and ended in the 1970s. In that period, from the 1940s to the 1970s, almost all of the key antibiotics that are used currently in the clinic today were discovered. And they were discovered using an approach that's referred to as the Waksman paradigm. Both of us are at the Waksman Institute at Rutgers University. The Waksman Institute was founded by Selman Waksman. He is the discoverer of the antibiotic streptomycin, which was the first effective treatment for tuberculosis, as well as a long list of additional antibiotics. And he discovered these antibiotics using the following approach, the approach that now is known as the Waksman paradigm. He collected samples of soil and grew from those soil samples, collected worldwide, bacterial cultures, and then tested the bacterial culture extracts for compounds that were able to kill other bacteria, were able to inhibit the growth of other bacterial species, working on the understanding that bacteria living in soil are in a resource-poor environment and must compete fiercely, tooth and nail, with other bacterial species inside the same soil samples, and therefore have been under pressure over eons to develop chemical weapons, if you will, to kill their counterparts. Some bacterial species develop chemicals, antibiotics, that they release into the medium to inhibit the growth of their competitors so that they can succeed in the competition for scarce resources. Other soil bacteria produce antibiotics, release them into the medium to kill other bacteria, which they then prey upon. And so Waksman realized that if one cultured these bacteria that produce and release antibiotics to inhibit the growth of their competitors or to kill their competitors and then consume them, these might be compounds that could be useful in human medicine. And this is how most of the antibacterial drugs in use today were discovered. They are compounds that went directly, without modification or with only trivial modification, went directly from bug to drug. That is to say, a compound produced by a soil bacterium to kill other soil bacteria that could be extracted and without modification or with trivial modification used to treat bacterial infections in a human.
Selman Waksman coined the term antibiotic, correct?
Yes, he did. So the word antibiotic is his term for a compound that's produced by a microbe to inhibit the growth of another microbe in dilute concentrations.
What ended this golden era of antibiotic discovery?
Eventually, researchers started isolating the same antibiotic again and again and again and not finding new antibiotics, meaning that the supply of easily accessible compounds that are produced in large concentration under many conditions by soil bacteria to kill other soil bacteria, and that would be suitable to be moved directly from the bug to the drug with minimal modification, minimal change, have been exhausted. The low-hanging fruit had all been picked. And that is the point when pharma began leaving this area. It was no longer sufficient to collect soil samples from around the world, culture them, and identify compounds in the soil samples, in the culture extracts of bacteria from the culture, from the soil samples that killed other bacteria.
And so how are you discovering antibiotics in your research?
So starting in, I would say, the 1990s and continuing through the present, techniques have become available that weren't available during the time of the golden age of antibiotic discovery. Techniques have become available to identify how the antibiotics work, to define the structures of the antibiotics when they are bound to their molecular targets inside bacteria, and to predict what modifications in a rational way could be made to the structure of the antibiotic to improve it. What this means is we now have the tools to be able to start with a poor candidate, a candidate that comes from a soil bacterium, but doesn't have the properties that would allow it to go directly from bug to drug. We have the ability now to understand how it works and use it as a starting point to tweak the compound so that it is able to be used as a drug. That wasn't available prior to the 1990s. That is available, but it's a much more complex process. It's no longer sufficient to find a candidate, patent the candidate, manufacture the candidate, and treat patients. It's now necessary to interpose a step after you've found a possible candidate to understand that candidate in detail, to understand its structure, its mechanism, its biology, and then to develop analogs, different chemical variants of the compound that will overcome deficiencies in potency or deficiencies in pharmacological properties and allow you now to use that, the variant, the chemical variant, allow you to use the chemical variant as a drug.
So then did the pharma companies rush back in now that there were new techniques, or?
No, unfortunately, the pharma continued to leave, and so pharma has abandoned this area, and that brings us to the discussion of the market issue. We've talked about how resistance arises, and I emphasize that when antibiotics are used in any context, in humans or in agriculture, that drives resistance. This gave rise, starting in the 1990s, or starting in the 1980s and 90s, to a move towards better stewardship of antibiotics as a limited resource. If, in fact, it's the case that when you introduce an antibiotic into the clinic, resistance begins emerging and will emerge and eventually render the antibiotic useless on the timescale 5, 10, 15, or 20 years, then one surely wants to limit the use in the clinic to those cases that are absolutely essential. You want to use a new antibiotic that's effective against resistant bacteria only against infections by resistant bacteria, not by general infections, and certainly not by non-bacterial infections. And that means restricting use. This in practice is accomplished by placing each new antibiotic that has the ability to kill resistant bacteria on a reserve list. And so we now have first line, second line, third line, and reserve drugs. If you develop a new antibiotic that is effective against every known pathogen, every known isolate of a given pathogen, including all of the most highly resistant isolates, that antibiotic will be placed on a reserve list. In essence, it will be put in a cabinet with a glass door in front, not to be opened, and only to be used in the exceptionally rare cases of patients who are resistant to all current drugs. And it will be kept on the reserve list until 5, 10, 15 years go by and the current first-line drugs lose effectiveness. And it will then be brought on as a third-line drug, where it will remain for another 5, 10, 15, 20 years until current second-line drugs lose their effectiveness, and it will be promoted then to second-line and eventually to first-line. What this means is that any company or any individual, any entity developing a new antibacterial drug effective against resistant pathogens will produce a drug that has no sales. In an environment where revenues are linked to sales, and that is our current pharmaceutical environment in the United States and in most of the world, developing a new antibacterial drug means no sales for decades. That means no sales during the period under which it has patent exclusivity, and often no sales at all under the time frame of a company and its planning or an investor and its planning.
Does that explain why we still have use of the antibiotics in agricultural in agricultural uses? That is to say, you mentioned that we're getting antimicrobial resistance because of antibiotic use in agriculture. The companies that are producing those antibiotics have a incentive to keep selling it to people that will provide it to their livestock, even if it's comes at a cost.
In general, in agriculture, we're talking about older antibiotics. No one in agriculture is using current reserve antibiotics in agriculture. So the compounds that are being used in agriculture are compounds that came from the golden age of antibiotic discovery. In some cases, antibiotics that still have related compounds in clinical medicine use, but in other cases, antibiotics that don't even have counterparts in use in clinical medicine.
So you're not that worried about use of antibiotics in agriculture?
Yes, I am, because I am concerned about the use of antibiotics in agriculture, because even if it's not the exact compound that's being used in humans, uh, use of any compound within a given antibiotic class in agriculture contributes to the emergence of resistance in that class and can set the stage for more rapid resistance emergence for specific compounds that are different from the ones used in agriculture.
Do you want to talk now about the policy
No,
around?
I probably should talk more about the broken market. So again, if you develop a new antibacterial drug that's effective against resistant pathogens, it will be put on a reserve list. It will not be used except in extremely limited emergency cases. After a period of perhaps a decade, it might move us onto the position of being a third-line antibiotic. After another decade, perhaps to second line. After another decade, to first line. But this means that revenues initially will be near zero. Revenues during the next decade or two will be very limited. This is an absolute barrier to interest from Big Pharma, is an absolute barrier to interest from investors. Big Pharma is looking for compounds that it can develop, market rapidly, put onto the market, and they become blockbusters with revenues of $1 billion or more per year per drug. They are not looking for compounds where they will invest $1 billion getting the compound to market and then find no revenues for the next decade and almost no revenues for the next 2 or 3 decades. The same is true for investors. Investors work on a very short time frame. They do not look at a time frame of decades. They look at a time frame of years. The last thing they would want to make an investment on is a compound that, if successful, will be restricted and unable to be sold. In order to overcome this market failure, it is absolutely necessary to break the linkage between revenues and sales. Currently, there's an absolute linkage between revenue and sales. Revenues come from selling compounds. In order to overcome the market hazard and the market failure with antibiotics, it's necessary to delink revenues and sales so that someone producing a new antibiotic, moving it through clinical trials, moving it through regulatory approvals, getting manufacturing in place, receives revenue for making that compound available to be put on reserve, and is given funding at that point that covers their expenses, possibly provides additional support beyond their expenses in the form of profits, but now has this compound in the cabinet, in the reserve space, ready to be used when it becomes necessary to be used. That's not a delinkage that has occurred in the United States. There have been experiments in delinkage in the United Kingdom and in Japan, but the United States is the real driver of sales, revenues, and policy in pharmaceutical development. Until the United States delinks antibiotic revenues from sales, there will continue to be a problem. There will continue to be no interest whatsoever from Big Pharma, no interest or nearly no interest from investors, and the only efforts that then are keeping the pipeline flowing in order to provide compounds to put into the reserve cabinet for future use will be coming from government funding and from charity, and will be carried out by small biotech companies and by academia.
So what policy changes would you recommend could occur if you were in charge? What would you
Oh,
recommend?
we absolutely have to move to a system of delinkage where an entity that produces a compound that has the required properties to be truly innovative and meet unmet medical needs immediately gets reimbursement for the cost of development. And then the compound is no longer needs to be the property of the developer. It now can become the property of the government. But that compound is available, the technology is in place, the technology has been developed, the technology has been transferred, and the costs of developing the technology have been reimbursed. When you do that, you do two things. You provide the necessary incentive to developers to proceed, and that they know their costs will be reimbursed. In addition, you ensure that stewardship will be in place, because if the government owns the property, intellectual property, and has the compound on reserve, the government has no incentive to overprescribe the compound, has no incentive to try to boost revenues by moving the compound into agricultural use. All of the negative incentives that currently exist when revenues are linked to sales disappear when revenues are delinked from sales.
Is this what that Pasteur Act hoped to accomplish?
Yes, for example. So there has been draft legislation, the so-called Pasteur Act, that has been introduced into each of the last several Congresses, has co-sponsors from both the Democrat Party and the Republican Party, but has never been passed into law. And it would do precisely this. It would establish that for new antibiotics that are brought to market, manufactured, there will be, and have true innovation and meet unmet needs, there will be reimbursement of the costs of development of that antibiotic drug
From the public, so
from the
taxpayer.
public. So it becomes then a public good. But so long as we work in the current market system does not drive toward public goods and will not develop compounds for reserve lists. It will not, in a system where revenues are based on sales, neither large companies nor investors will develop compounds that will have no sales if they are truly innovative and useful, because those will be precisely the compounds that will be put on a restricted list, on a reserve list.
Why wasn't there more support for the Pasteur Act? What are the arguments against it?
The argument is that this would provide a gift to the pharmaceutical industry. The pharmaceutical industry isn't even in this space, so that's not really a logical response. But the thought is that a cash reward to a pharmaceutical developer upon development of a new compound is a gift to pharma. And there has been strong opposition from many in the Democrat Party that this is a gift to pharma. There has been opposition from many in the Republican Party that this is an interference in markets.
And you have had you haven't been able to win over any of these lawmakers?
With the Infectious Diseases Society of America and with the Pew Foundation, I participated in many congressional visits, both in New Jersey, congressional offices of our senators and representatives, and in Washington, D.C., in congressional offices. Congressional staff seem to be very attuned to the issue and understand the significance of the issue and respond, but we can't. But the senator can't support this because it would look like a gift to pharma.
So optics over potential.
Optics.
Yes.
Optics.
Well, I guess one question though is there are alternatives. I guess one alternative to antibiotics is phage therapy. Do you have anything that can you talk a little bit about phage? I know that's not what you're working on,
No,
but
that's not what we're working on. But so phage therapy refers to using viruses that infect bacteria as tools to treat bacterial infections. This is a very old concept, predates antibiotics by at least 2 decades. There have been limited cases of success where individuals who had what are called pan-resistant bacterial infections. That is to say, infections resistant to the full set of currently available and approved drugs, even those on reserve, in some cases have been successfully treated with phage therapy. But phage therapy has two issues that I think will keep it always a niche therapy. The first is that it is extremely narrow spectrum. And when I say extremely narrow spectrum, that means not only is it not affecting multiple genera of bacteria or multiple species of bacteria, it frequently targets just a specific strain of a species of bacteria. That means it cannot be scalable. It never will be scalable. It never could be used as a first-line treatment the way that antibiotics are used. And it will always be challenging to even apply it on a limited scale basis. Second major problem is that resistance arises, and resistance is even faster to arise than with antibacterial compounds. I told you before that the easiest way for resistance to an antibacterial compound, an antibiotic to occur, is for the bacteria to acquire a mutation that alters the binding site, the molecular target for the drug. In the case of phage therapy, the molecular target for the phage is the receptor on the outside of the bacterial cell that the phage binds to first interact with the bacterial cell. The interfaces between a phage and its receptor are very large. That means the number of residues that could be mutated to give resistance is very large, which means that the rates of emergence of resistance are enormous. So for a typical small molecule antibiotic, an antibiotic drug, the rates of resistance are on the order of 1 in 100 million per generation to 1 in a billion per generation. For phage therapy, for a phage bacterium interface, the rates of resistance are 1,000 times higher than that. And so resistance erodes the usefulness of antibiotics, but it erodes the usefulness of phage therapy almost instantly. You get resistance in an individual patient. One patient has more than sufficient bacteria to have resistant bacteria.
But as a platform, could one argue that the phage therapy is a bit more adjustable and fluid because you can program it? You can control the genome of the phage and you can introduce things.
You have all those tools, but you have the challenge that it is inherently extremely narrow spectrum, extremely high resistance emergence. It also is a one-time therapy, or can be a one-time therapy, because the patient will develop an immune response to the phage, not just to the specific strain of phage that are used, but to the entire category of phage represented by those used.
What about the use of AI, Richard, in helping to identify, new antibiotics?
There have been papers, but the AI doesn't really have the ability to address key limiting points on the drug development pathway, at least not for antibacterials. So I think that will be something that receives attention when it is successful, but it rarely will be successful.
Is there, I did want to get into the, issues around dangerous research, dual use research. Obviously, I just mentioned phage, and there are concerns about synthetic viruses. These phage are bacterial viruses, so at least right now, there doesn't appear to be any, imminent threat of development of bacteriophage for dual use purposes. But I wondered, are there any concerns about the creation of superbug, antibiotic resistant bacteria that could be used as biological weapons?
The threat there would come with those bacteria that historically have been used as biological weapons, enhancing them to make them pan-drug resistant to all of the available therapies. So it is actually relatively straightforward to take a bacterium and engineer it for resistance, select resistant variants, or introduce genetic modifications that in a planned way, provide resistance. If one took a bacterium that historically had been used as a biological weapon, like the anthrax bacterium or the plague bacterium, the glanders bacterium, the melioidosis bacterium, and introduced multiple drug resistances, that would increase the threat level.
But how would that compare to a virus that was transmissible by air, through
Well,
air?
in the case of the anthrax bacterium, that's a bacterium that can be distributed as a spore powder, but it does not transmit efficiently or at all effectively from human to human. So it's an endpoint illness that makes it actually useful as a biological weapon in some contexts, but it is not something that can cause a global pandemic. It doesn't transmit from person to person. Of the bacterial species that I mentioned, the only one that is a potential pandemic pathogen is the plague bacterium, Yersinia pestis. That is a pathogen that can transmit efficiently human to human, can transmit even by a respiratory route human to humans. That is a bacterium that one could make into a highly dangerous pathogen by making it pan-drug resistant. The Russians, under the former Soviet Union, were very actively involved in making multi-drug resistant Yersinia pestis. Again, that's a relatively straightforward thing to do.
Okay, I guess I'll ask the follow-up then. Are you concerned at all about these as, modern day biological weapons, or are you more concerned about the viruses?
I'm much more concerned about viruses, as we all have had the opportunity to see over the past 5 years. A pathogen that's transmissible human to human, particularly one that's transmissible by an aerosol droplet route, or even worse, an aerosol route, is a much greater concern than a pathogen that requires contact.
I did want to ask you, do you want to comment on the use of animals in research to identify new antibiotics and test antibiotics? And I guess this is a very polarizing issue, and I know that we talked about how we have to sort of find a balance between use of animals for the benefit of humanity and torture.
For antibiotic drug development, it is inescapable. It is not possible to develop antibiotic drugs for use in humans without access to a vertebrate animal model. That vertebrate animal model is a rodent model. That means a mouse model, sometimes a rat model. But without the rodent model, one simply cannot rank compounds. One doesn't even have remotely the ability to rank compounds and decide which compounds are worthy of being pursued. None of the experimental methods that allow us to look at interactions of candidates with their target molecules or interactions of candidates with bacterial cells in culture give a readout of what the efficacy of the compound will be in a vertebrate animal. We have in antibacterial drug development, we do have the advantage that the target that we're aiming for is a bacterial cell. And that's the same target, whether it's in a culture or in an animal or in a human. And so we have much more translatability of information from culture to animal to human than in many areas of therapy where the target is a human cell, which is different from the vertebrate animal cell and very different from that cell in culture. So you do have more translatability, but still you cannot rank compounds because the efficacy of the compound depends not only on how well it engages its molecular target, it depends not only on how well it kills a bacterial cell, it depends crucially on how the compound enters a vertebrate animal, how it is distributed within the body of the vertebrate animal, how it is metabolized within the body of the vertebrate animal. And there are no means. There are no computational means. There are no small molecule or cell culture or organoid models that provide any useful information in making that prediction. So we always say that we can develop our list from work in vitro, that's work in biochemical systems and work in culture, we can develop a list of our top 40 candidates. But then to rank those 40 candidates, our standard answer is only the mouse knows. The biochemical experiments in the culture simply can't tell us. They can't even make reasonable predictions. But a simple experiment of testing efficacy in a mouse model rapidly identifies which are the top 2 compounds out of the top 40.
So just to be clear, you're talking about using mice or rats?
So the primary efficacy model is mouse. And so the current developmental pathway calls for establishing efficacy and pharmacokinetics and pharmacodynamics in mice. Optionally for obtaining that same information in rats. And when I say optionally, that means in order to get FDA approval to move into clinical trials, to get what's called an investigational new drug approval from the FDA, you absolutely require efficacy, pharmacokinetics, and pharmacodynamics in mice, and you'll require efficacy and pharmacokinetics in rat. To get that investigational new drug approval, in addition, you will need safety studies and pharmacokinetic studies and safety studies in larger vertebrate animals. Conventionally, that today is done using either dog or non-human primate. That's an area of drug development where the rules come not from the science, but from the FDA. And that's an area of vertebrate animal use that I think could be very much trimmed back.
You think it's essential that we maintain use of mice or rodents?
You simply can't. You can't develop an antibacterial drug or any drug without a mouse or rat model, without a rodent model. And to move beyond that, in order to be able to predict effective doses in humans, you also would need one additional pharmacokinetic data. That's information on availability and distribution within the body of the vertebrate animal for one larger vertebrate animal. I would recommend that the FDA change its current policies, which require rodent, dog, and or non-human primate to be rodent mini pig, so swine. And with 2 rodents and 1 larger vertebrate, you now have 3 different endpoints on 3 different data points on animal size, which is enough to do allometric scaling and predict reasonable doses in humans.
So the results of those experiments are informative about what happens in the human.
Yes. And so what the key point is in order to define your best candidate, there is no alternative but to use a rodent animal model. In order to define what is the pharmacokinetic pharmacodynamic driver, that means the parameter of availability that determines efficacy, which can be for some compounds the maximum dose that's achieved, called Cmax. For other compounds, it's the total dose that's achieved, called AUC. And for other compounds, is the time at which the concentration in the blood plasma is over the efficacious level. It's called time dependence. In order to define which one of those 3 parameters is the driver, you must have an animal model. That information can be obtained in rodents as well. So the rodents can tell you the efficacy, which you must know. The rodent information can tell you the pharmacokinetic, pharmacodynamic driver, which you must know. In order then to make even a guess as what dose you will need in humans, you need at least 3 different animals on the size scale. And that means mouse, rat, and something larger than rat but smaller than humans. And with the data in 3 of those and knowledge of the pharmacokinetic pharmacodynamic driver, one can scale to the dose level that would be appropriate for a human trial. Without that, you are blind, and there isn't a substitute. So I think non-human primate studies could be removed, could and should be removed by the FDA from the requirements for an investigational new drug approval. I think studies with a companion animal like dog could and should be removed, but there does have to be one intermediate vertebrate animal. We eat ham, we eat pork, we eat bacon. Pig is reasonable.
Okay, thanks for. I do appreciate you going through that because I'm certainly that's something that very few scientists, at least in my opinion, are addressing right now. And there's a lot of outcry in the public about the abuse of animals in research, and I think it's important for scientists to have that conversation head on.
The biggest use and the main use of larger animals is for this so-called IND enabling study, the Investigational New Drug Approval Study. And that is determined entirely by policy. The current FDA commissioner, Marty Makary, has said that he is looking toward scaling the requirements down. I believe that an enormous amount of money could be saved, an enormous amount of time could be saved, and animal studies, safety and toxicology studies in larger animals could be avoided simply by saying that 3 animal species is sufficient, and suggesting that those 3 be mouse, rat, and mini pig.
You don't have a large investment in a mini pig company, do you, Richard?
No,
Yeah.
we haven't. We only have worked with mice, but we are at the stage in drug development for our most favorable compounds. We are at the stage in drug development known as candidate selection, where we are naming which of the more than 800 compounds in that series we synthesized and analyzed. We're identifying which one is our candidate, which is our backup. That gets followed by candidate de-risking, and within a time frame of 6 months to a year, we will be entering that phase of the IND enabling studies, the studies that require moving to additional species for efficacy in a second species, which would be rat, and pharmacokinetics in at least 2 additional species, which would be rat and pig.
Is that you said you have a company now, right? That's sort of on
So
paper.
in June, in June, we formed a startup called APY Therapeutics to develop the class of compounds that are most promising and most advanced from our research, which are compounds called aryl mixoperonins, or APYs, hence the name of the company, APY Therapeutics. We have synthesized and analyzed more than 800 compounds in this series. We have 3 compounds that we currently are in the final stage of ranking. That final stage of ranking involves confirmatory dose escalation mouse infection studies. And the studies are happening literally at this moment. By the end of October or beginning of November, we will be able to make our selection of our preclinical development candidate and backup. So what are APYs? As I mentioned before, we start with the structure of RNA polymerase and identify useful potential new drug targets within RNA polymerase, perform screens to identify compounds that inhibit RNA polymerase through those targets, and then characterize the compounds. One of the classes of compounds that we analyzed are so-called mixoperonins. Mixoperonins are compounds that are produced by soil bacteria. They're produced by a group of soil bacteria that are predatory soil bacteria. They prey on other bacteria. And the way they kill the other bacteria is they synthesize mixoperonins, release them into the environment. That kills other bacteria, and then the producers of the mixoperonin consume the cellular debris of the killed bacteria. Mixoperonins are produced by bacteria, but they do not have the properties to go directly from bug to drug. Unlike the compounds that were developed in the golden age of antibiotics, mixoperonins have poor aqueous solubility. They dissolve poorly in water. They have poor metabolic stability. They do not have the pharmacological properties that would allow them to be used as a drug. They also have insufficient potency to be used as a drug. They were discovered in the 1960s and 1970s in the golden age of antibiotics, but because their properties did not allow them to go directly from bug to drug, they were abandoned. We began studying them as some of the earliest compounds we started studying when we began our work on antibacterial drug development around 2000, 2001. We were able to show how they killed bacteria, that they did so by inhibiting RNA polymerase. We were able to determine the structure of a mixopronin bound to RNA polymerase, so we could see exactly how it bound. And we were able to use the structure to predict how we could change the chemical structure of mixopronin to make it better. And as I said, we've made more than 800 of these compounds, rationally guided, structure guided, rationally designed derivatives designed to overcome the limitations of the product from the bacterium. That is to say, to make it more potent at binding to RNA polymerase and inhibiting bacterial cells, to increase its water solubility so you could dose at higher levels, and increasing its metabolic stability so that it would persist inside an animal and be effective. We were able to develop variants of the mixopronins that we call aryl mixopronins, or APYs, that have an order of magnitude increase in potency, a multiple order of magnitude increase in aqueous solubility, and an order of magnitude increase in metabolic stability, and are orally available, which means that they not only are effective if injected into an animal, but also if taken by mouth. And these are all characteristics that were absolute minimum thresholds for developing an antibacterial drug in the current market context. And we've been able to identify aryl mixopronins that have very high efficacy in mouse models of infection against lung infections, pneumonia, as well as also against skin and soft tissue infections. And we're able to show that our best compounds are effective against most of those 8 types of bacteria I mentioned earlier that represent the greatest burden. They're effective against mycobacteria. They're effective against streptococci. They're effective against staphylococci. They're effective against enterococci. They're effective against Acinetobacter. And most important, or perhaps most important, they're effective against all known isolates. There is no identified clinical isolate that is resistant. So there is no resistance in the community, in the clinic, globally to these compounds. So they represent an entirely new class of compounds with a new structure, a new mechanism, no pre-existing resistance. All of these are positive features, but it means if we do develop them, they go straight to the reserve cabinet.
So you're just doing this out of the good of your heart.
Doing this because I have always wanted the research that I perform to have translational impact. And so, as I said, we started about 2.5 decades ago, beginning translational work. The aryl mixopronins are our closest compound to moving out the other end of a pipeline.
How many employees does your company have?
We have 3 employees, none of whom is paid. So we have 3 uncompensated employees.
Right. I wanted to
the 3.
Can you clarify that we're not talking about this large scale operation?
Oh no, this is headquartered in my home. I am 1 employee. My wife is director of chemistry.
Okay.
And a researcher from my laboratory at Rutgers is director of microbiology. And we are all doing this on an unpaid basis. Again, in this space, this is not an area of research which one pursues for profit or for income. One pursues this because there is an unmet need. And you earlier asked about patents. We have perhaps 10 patents on the aryl mixopronins. One has patents because without the patents, it is impossible to advance the compound through the stages of drug development. It's not something that you expect any revenue ever to flow from.
I think a lot of people are surprised at how much you're doing outside of Twitter, right? Given how you're able to juggle all these things and I think that's great that you're talking about the antibiotic work that your lab does. And I wanted to end, Richard, with something that we've talked about a lot. Why is Waksman Institute not that well known, even at Rutgers?
Well, I think part of it reflects the fact that Selman Waksman is no longer with us. And when Selman Waksman died a long time ago, and when the people he had hired who worked in antibiotic development retired and then died, all of this was near the end of the golden age of antibiotics. There had been no activity following that on antibiotics until I began work on antibiotics around 2000. And so when I arrived at the Waksman Institute in 1987, there was only one lab doing antibiotics. That closed shortly after my arrival. There was a long gap period, and the focus of the institute moved elsewhere, and the recruitment of new faculty in the institute moved elsewhere.
But now that it's become an urgent public health threat, doesn't it make sense for there to be some investment in the Waksman Institute?
Well,
Either
I obviously would welcome that.
I guess, but what form would that take? It seems that what you're describing is policy is a major barrier to getting anything done. So wouldn't it make sense for Waksman? I mean, a leading question, of course, but what would you say if you, and by extension me, of course, you and I start a policy center, a science policy center at Waksman that's focused on both antimicrobial resistance and biosafety? Why wouldn't that be successful, Richard? Explain to me why that wouldn't be the most successful policy center in the world.
Well, that's quite an objective to be not merely successful, but the most successful in the world.
But the bar is low, I think, correct? I mean, think about biosafety policy, right? All of those think tanks, tweeting is more effective than those think tanks with those dozens or tens of millions of dollars that they get, right? Couldn't we do the same thing with antimicrobial resistance?
Possibly.
You don't seem that enthusiastic about it, Richard, I guess.
I would welcome it, but I don't see the resources at hand that would be necessary to make it happen.
Well, we'd have to solicit private donors. But if there was a private donor that wanted to invest in the Richard Ebright Center for Biosafety and
I would
Antimicrobial,
welcome that.
you'd welcome that?
Of course.
Okay. Well, I guess I'll end with that. Is
I
there
don't have any up my sleeve.
Right. Well, I get you are working for free with this company. So at
Yes,
least
yes. The work that we do with APY Therapeutics is people can afford that price.
the price is right.
The price is right. Yes, the price is
Yeah.
right.
Richard, do you have any final words before I end this podcast?
The current antibiotics, our current first-line antibiotics, will be gone within 2 decades. Our current second-line antibiotics will be in the process of having their utility eroded by resistance and will be gone a decade after that. That is why we need the reserve antibiotics. We constantly must have reserve antibiotics waiting to go up to third line, then to second line, and finally to be promoted to first line antibiotics. If we don't develop new reserve antibiotics, then 2-3 decades out from today, we will have an instant crisis. We will be off the end of the cliff, and almost all parts of modern medicine will become unavailable. Unfortunately, the market failure makes that a real possibility. To avert that possibility, we need efforts like the efforts I describe in small companies and academia that are pursuing this not for revenues, but to meet an unmet need. And we need ultimately to delink sales and revenues.
All right. Well, Richard, thank you very much for doing this. I'm really glad that we got to talk about something that's much less controversial than what we normally discuss. Take care. This has been Bryce Nickels on Science from the Fringe.